All right, folks, let's focus on graded potentials. Now, graded potentials are always going to be postsynaptic potentials. So let's orient ourselves. If we're talking about a neuron, we have the dendrites and the cell body or soma, and that's where graded potentials are going to occur. The axon is going to be where action potentials occur. And of course, the message is going to go to the axon terminals. And these axon terminals can be connected to muscles or, of course, other neurons as well to, again, send the signal. So when we're looking at this photograph here, we have our cell body and our dendrites, and we also have and those will be our post-synaptic, so they're going to receive the message. And then you see that on different parts of the membrane, we have axon terminals, which are going to be our pre-synaptic, and they're going to be sending the signals. For this green area here, and that's going to be the area where you're going to have the strongest signal. Okay, that's going to be the strongest signal, strongest stimulus is right where you're at that membrane. And of course, that stimulus can dissipate as you go along and you get to the trigger zone, or again, the initial segment. You can also call this the axon hillock. Okay, so let's go ahead and get into it. Now, postsynaptic potentials mean that you're going to have some kind of change in membrane potential at this terminal here. And there's two types of graded potentials that you can have. You can have a graded potential that's going to depolarize. And we know if it's going to depolarize, we have more of a chance to get to that negative 55 millivolts threshold, right? To start an action potential. And those are called excitatory postsynaptic potentials, or EPSPs, okay? This is going to make the membrane more positive. And what you normally see is that Na plus or sodium therefore gets to go inside the cell. If it goes inside the cell because it has a positive charge, it's going to make the cell more positive. Make the membrane more positive, and of course inside the cell more positive. Now, we also have different graded potentials that will cause an inhibitory effect. Those are called inhibitory postsynaptic potentials, or IPSPs. This is going to make the membrane more negative. So if sodium comes in and makes the membrane more positive, we're thinking depolarization if we're going back to our terms. If we have the inhibitory postsynaptic potentials, say if you have something like chlorine coming in, which is also already has a negative ion charge, it's going to make the membrane more negative, and of course inside the cell more negative, and that's going to be creating more hyperpolarization. Okay? And what do we mean by the hyperpolarization, and what do we mean by the depolarization? Remember, we're starting at negative 70 millivolts because our membrane is at rest. And if we have the membrane becomes more positive, we go to around plus 30. If it becomes negative, then we can go as low as negative 90 or even 100 millivolts. So just keep those things in mind. Okay? So there's two types of graded potentials that you can have. And remember, each of these graded potentials can have a certain strong stimulus or a weak stimulus. Either way. Let's get into an example of the sequence of depolarizing. That's going to be a big focus, is depolarizing. Yes, you can have the hyperpolarization, but our main focus is going to be the depolarization. So we're talking about excitatory postsynaptic potentials. First, the gated sodium plus channels are open in response to what? A stimulus. So those neurotransmitters are coming out from these axon terminals, and they're going to, of course, bind. Those little ligands are going to bind to a receptor on that ligand-gated channel. And of course, you're going to have sodium, let's just say this is sodium here, rush into the cell. Okay? So sodium ions are going to enter into the cell. That's going to make the inside of the cell more positive. So if it's more positive, it's going to be depolarized. Okay? Then the depolarization is going to spread. Now here's where the work comes in. Let's say we come in and this first initial stimulus signal is going to be, let me get black. It's going to be like negative or like 10 millivolts. 10 millivolts, right? As we know, this is going to be greater here where the initial segment is, or not the initial segment. It's going to be greater here where the stimulus is. That 10 millivolts is straight up going to be 10 millivolts because we have the neurotransmitters connecting to the ligand-gated channels, and sodium keeps coming in, right? But as we move down the cell body, keep moving down the cell body until we try to get to the initial segment, guess what? That amount, those 10 millivolts, is going to start to get lost. It's going to dissipate. So instead of having 10 millivolts here, Maybe you'll have 5 here. Maybe you'll have 2 here. Maybe by the time you get down to the initial segment, it's only going to be 1 millivolt. And that's really not going to be enough to hit that negative 55 threshold. So let's go ahead and see an example. So let's say, of course, we're all starting at negative 70, which is going to be resting membrane potential. We have a stimulus that comes in and we're like, yes, we got sodium coming in. That means the membrane is going to be more positive. We're about to get depolarization. And our goal is to hit that negative 55. Well, when you first have the surge of sodium come in, you might get to like negative 60. But over time, that particular stimulus will dissipate and you'll go back to resting potential. Okay? So, of course, the big question is, how do we get to the negative 55 if these single graded potentials are going to dissipate? There's the graded potentials, they start having an additive effect. They add each other up, and we'll talk about that next. But before, some folks might ask, well, how do we go from 10 millivolts? Why is it not just 10 millivolts as we're going through the actual cell body? And that's a great question. So if you're going from 10 millivolts, if you're going from 10 millivolts and you're like, well, how did I get to 5? I don't understand that. 10 millivolts and you're going to 5 millivolts. The reason why you actually are going to have some of that sodium and that sodium signal loss is because of leak channels. So we got stuff just fighting against this, right? Here, at this membrane portion here, where you're going to have the synaptic cleft, as we talked about with synapses, yes, you are going to have a rush of sodium coming in, right? Rush of sodium coming in. But if I have a leak channel here that doesn't need a light, and a leak channel here, sodium's also going to be going out because, again, we are increasing the concentration of sodium inside the cell. So when it comes to leak channels, they go off of diffusion. It's going to go from high to low. So some sodium is going to go out. And because some sodium is going to go out, you are going to decrease the overall signal. So that's the reason why. Let's go ahead and get into how each of these individual graded potentials can be added up to actually get to that threshold. So that is going to be a process called summation. In other words, the EPSPs and also the IPSPs are tiny. And alone, they don't have too much impact. But guess what? They're going to be rarely alone because you have thousands of other neurons that all send postsynaptic potentials. And we can even go back to see that. We have a lot of neurons here that are going to be sending those postsynaptic potentials. We have the axon terminals here creating that synaptic cleft to be able to make sure that we're getting enough sodium in. Okay? So just keep that in mind. Now, the only way that we can get to that threshold is if we have what's called summation. That's going to be adding multiple postsynaptic potentials at the initial segment. Now, how do we do that? There's two different ways. There's going to be, temporal summation and spatial summation. Now, temporal summation is all about one neuron talking to our neuron or another neuron. And they're going to end up overlapping in time. So, for example, you have one neuron that's going to fire. Okay? So you're at resting potential. It's going to fire. And it's like, oh my gosh, I'm trying to reach negative 55. Okay, I'm going to dissipate. But before it completely goes all the way back to rest, there's another one of that same neuron that's going to fire. And so what happens is it kind of catches it. Then it's going to keep going and depolarizing. Then this one's going to start dissipating, and then you're going to have another of that same neuron come in until you reach the negative 55 threshold. Once you reach that threshold, action potential. Action potential. And this is going to be temporal summation. All right, the other type of summation is going to be spatial summation. And think of it, spatial, spaced out. It's basically a bunch of synapses that are super close to one another. And so they're on one huge chunk of the membrane. They're super, super close. So let's go ahead and get an example of that. We have our axon terminals here. With the temporal summation, you're going to have the same axon terminal going, presynaptic and postsynaptic neurons coming together to just keep on giving us a signal. When one goes to start to dissipate, another one comes in, same signal, right? But with summation or spatial summation, you're going to have a bunch of them sending different signals, all in one chunk of the membrane. So let's say we even had another terminal here, another terminal here, as many terminals as you need, okay? Now, when it comes to spatial summation, a lot of ways it can happen. You can have all of the spatial summation happen with a bunch of EPSPs near one another, a bunch of IPSPs near one another, or both EPSPs and IPSPs right next to one another. So we're going to get an example of when both of them are happening. So say we first get an EPSP. We're like, yes, we're at our negative 70 membrane. We're going to go, ooh, we're going to go and try to get to that negative 55 millivolt threshold. And then guess what? We're going to have an IPSP that inhibits it. So if the IPSP comes in, we're going to hyperpolarize and go back to rest. But then you might have another EPSP because, again, everything's right next to one another. Come back in and you'll get closer to that membrane, that negative 55 millivolts for the membrane. And then here comes another IPSP, so you get lower. Then you might have two EPSPs come in. Boom. When this dissipates, another one comes. And then, of course, you might have another IPSP come in. Then finally, you might have 3 different EPSPs come in, which allows you to basically get to that negative 55 threshold, and therefore you will have an action potential. Okay? So when it comes to temporal summation, just think it's going to be greater potentials at one synapse overlapping in time. So that one single synapse that we talked about here. And we start erasing a couple things too. Okay? So we want to focus on this synapse here. Temporal is going to be that same one. So yes, we'll have one come in, and then it'll dissipate, another one come in, dissipate, and then of course you hit that negative 55 threshold. And remember, this is going to try to build it all until you get to that initial segment, because this negative 55 is happening at that initial segment. That's what we're trying to get to. Okay? So remember, it's always going to be at the initial segment. I'm showing you where it starts because that's where the synapse is showing us, but remember, you can also have it in this area, and other areas that are closer to the initial segment as well, because this is where the dendrites are. Now, spatial summation, let's see, we'll go back to the same photo. It's like if we were focusing on, let me erase this, all of this synapses, all these axon terminals. Let's just add some more here. Because they're so close to one another, one could be an EPSP, one could be an IPSP. We don't know. Okay? But what we do know is, depending on how many we have, yes, we might have some hyperpolarization, depolarization, but our goal is to get to that negative 55 threshold at the initial segment. And again, this could be happening in other areas of the membrane because we're talking about thousands of neurons. We're not saying that's just always going to be in this one little chunk. You have a chunk here, chunk here, chunk here, chunk here. Just keep that in mind. All right, folks, so that is going to be our understanding of graded potentials. Remember, the strength of the graded potential it's important, right? When you're talking about graded potentials in general, you're like, okay, if I have a weak stimulus, it's probably not going to get to the initial segment. But even if you have a strong stimulus, it's probably still not going to get to the initial segment. You have to make sure you're adding or summing these multiple EPSPs and IPSPs together in order to get to that negative 55 threshold. Let's go ahead and get into a worksheet. Number 14, it says, imagine 15 neuron synapse on one postsynaptic neuron. So we're talking about 15 neuron synapse on one postsynaptic neuron. At the trigger zone, 13 of the neurons produce EPSPs of 2 millivolts each, and the other 2 produce IPSPs of 3 millivolts each. Okay? The threshold for the postsynaptic cell is negative 55 millivolts. They want to know, in this scenario, would an action potential be produced? That means will we be able to reach that negative 55 millivolts threshold? The postsynaptic neuron has a resting membrane potential of negative 70, which is what we normally know about. This little chunk, right? So let's go ahead and break this down. 15 neurons synapse on one postsynaptic neuron. So I have my little postsynaptic neuron here, and I'm saying that 15 neurons, or those terminals, are going to synapse, okay, on one postsynaptic neuron. So yes, we definitely are talking about spatial summation. And it's saying at the trigger zone, so let's go ahead and write that trigger zone here. These are axon terminals. 13 of the neurons produce EPSPs of 2 millivolts each, and the other 2 produce IPSPs of 3 millivolts each. So that means if we're talking about something that's spatial, you can have IPSPs and EPSPs, right? That would be my first little indicator. Now, if you're talking about temporal, you can have all EPSPs or all IPSPs. But when it comes to spatial, you can have both, right? So that'll be another little indicator of just saying, okay, I'm definitely talking about spatial. Now, let's get into the math here. 13 are going to have neurons that produce EPSPs of 2 millivolts each. And then we're going to have 2 that are going to produce 3 millivolts each. So what can we do? We can definitely multiply those, right? We're talking about summation. We got to make sure we can add this stuff up. So this is going to be 26 millivolts. Okay. And remember, this is definitely going to be positive. And then we're going to have 6 millivolts when it comes to IPSPs, and that's going to be negative. Why? Because remember, IPSPs are going to cause hyperpolarization. EPSPs are going to cause depolarization. So let me make sure. No. Oh man. Okay. Hold on. I'm back. So here's all of our EPSPs and IPSPs. We got the numbers here. Now we got to figure out in total what is going to be the change between the two. So we just subtracted 2. 26 minus 6 is going to give us 20 millivolts. So it's 20 millivolts that we're working with. Okay. And what that means is that's going to be the change in the membrane potential. What are we starting off with? We're starting off with negative 70 millivolts. And then we're going to add 20 millivolts. Why is it positive? Because remember, we had more EPSPs than we had IPSPs. So negative 70 plus 20 millivolts, which is going to be the change in membrane potential we have due to our EPSPs and IPSPs, is going to equal negative 50 millivolts. So we got our math down. Now the next question is, can we produce an action potential? Well, we know we have to hit a threshold of negative 55 millivolts. This is even a little bit more positive. So yes, we will have an action potential. All right, folks. The next video is going to be about action potential. So what a great segue. See you there.